Diflubenzuron is this particular MRL at the request of the CCRVDF is considering specifically residues in Atlantic salmon. So I would say universally all Atlantic salmon is farmed, and sea lice, which is an ectoparasite of fish in fish farms, is a real problem in terms of reducing overall harvest of the salmon. So we've provided a little bit of information on exactly what it is. It's an insecticide. It's a relatively good insecticide because primarily it's inhibits the formation of chitin, which is a principal component of exoskeleton. So just by this fact, it would be expected to have a relatively low toxicity as compared to mammalian species who don't rely on chitin formation. The other issue, as with a lot of veterinary drugs, it already has a number of existing pesticide-approved uses, and there's a large number of MRLs available for other products worldwide. So we haven't provided details of this, but in most MRL databases, you will find Diflubenzuron that is MRLs being available both in, say, cereal grains, fruit crops, and food-bearing animals. So this pesticide or insecticide has been approved for quite a number of years, but there was an issue that came up, and this is relatively recently, that the use of the pesticide may be associated with a potential genotoxic metabolite of 4-chloroaniline, or PCA. So as you might expect, there is limited acceptability from a consumer perspective to know that carcinogenic substances are being deliberately added to food. So in this case, even though the carcinogen is not being directly added, it's thought that the either as a degradation product or a metabolite of diflubenzuron, this 4-chloroaniline may be produced. So the Codex Committee, Residues of Veterinary Drugs and Food, requested that GFoRSS revisit its previous decision of approval of diflubenzuron, and whether or not there was sufficient information to suggest that this potentially genotoxic metabolite or degradate could be produced. So GFoRSS did this, and in their 81st meeting, they agreed and said that this 4-chloroaniline could be produced. The evidence to date suggested it was genotoxic, and therefore they were unable to establish an ADI for diflubenzuron. Even though JMPR, which is another association that looks at pesticide approval, had already developed an ADI for diflubenzuron. But this is from a number of years ago, and in their assessment, they did not consider the potential that there may be 4-chloroaniline exposure. So similarly, at the same time, the European Committee that looks at veterinary drugs and EFSA also looked at diflubenzuron. And so and they basically came to the same position that it's possible that the use of this pesticide or insecticide, and in this case, veterinary drug, could be associated with exposure to the 4-chloroaniline. So in its most recent assessment at the 88th GFoRSS, the decision was once again to consider potential exposure to this 4-chloroaniline. So GFoRSS took the position that they would look at the actual mechanism of action for this particular chemical. And it should be pointed out that in the initial pesticide approval for diflubenzuron, none of the chronic bioassays that were conducted in experimental animals gave rise to any concern about cancer. So this was something that was picked up later on, and it was indicated that there were available animal bioassay studies with 4-chloroaniline that did suggest it was positive for producing cancer. So what GFoRSS did in its 88th meeting, when it was requested to look at potentially developing an MRL for diflubenzuron in salmon, was to, as we mentioned here, look at the mechanism of action, how cancer may develop 4-chloroaniline, and they came to the conclusion that this was not a direct DNA reaction product. It was something that was an indirect mechanism through oxygen radical generation, which is a relatively well-known process where cancers or tumorigenic responses can be developed with lifetime exposure. And therefore, they concluded that any cancer seen with 4-chloroaniline was likely a threshold. So, and therefore, you could define a dose where there would be no significant increase in cancer. So, following that decision, they went about to establish an ADI. They reversed the decision from 2015 and said they could establish an ADI for diflubenzuron. And this was the same value that had been set previously by the JMPR. So, and just a bit of background history that we provided, that the value that GFoRSS came up with for diflubenzuron is the same that was established by the US Environmental Protection Agency when their pesticide registration for diflubenzuron was considered. So there's a number of different ways that you can assess exposure to potential carcinogens. One of those is to develop a separate health-based guidance value for the carcinogen, which GFoRSS concluded was not appropriate because of lack of information. And the second method that they used was they looked at the estimated dietary exposure for 4-chloroaniline from use of diflubenzuron and compared it to these. For those who may be familiar, there's a threshold of toxicological concern approach where there's an entire science behind development of the threshold of toxicological concern, where hundreds of chemicals have been categorized based on their structure into various categories. And these are classes of chemicals, class 1, class 2, class 3. And what they do is assign a particular value that if the exposure is less than these values, there is unlikely to be a health concern. So in this case, GFoRSS assigned 4-chloroaniline to the threshold of toxicological concern class 2, which has a value of 1.5 microgram per kilogram body weight per day. So it just means that in hundreds of chemicals that are structurally related to 4-chloroaniline, all of them have toxicity that is 1.5 microgram per kilogram body weight per day or greater. So any exposure less than this value is unlikely to be a health concern. So in a similar process for the flormethrin, GFoRSS also developed estimates of chronotary dietary exposure. In this particular case, GFoRSS declined to set an acute reference dose, which simply means that the acute toxicity associated with this chemical is too low to consider single or acute oral exposures. So their chronic dietary exposures were considered, and that's based on taking into consideration all fish/salmon consumption that has been reported or available from national estimates of dietary exposure. And they came up with, based on the ADI that they proposed of the 10 microgram per kilogram or 10 parts per million, that there would only be 14% of the ADI would be realized. So these are also looking at very elevated levels of fish intake on a chronic basis, and application if considering that all salmon contained the diflubenzuron at 10 parts per billion. So in reality, they did have because diflubenzuron has been approved for use in a number of countries, specifically for this purpose. It's just that GFoRSS has never elaborated an MRL specifically for salmon. So in all the available surveys that have become more prevalent, because there is sort of an environmental issue to the use of these sea lice preventing compounds in veterinary medicine, specifically for aquaculture. They said that the vast majority of results for diflubenzuron in salmon were less than 2 parts per billion. And in fact, a very large percentage had no detectable residues, once again using LC-MS-MS, which has typical detection limits of 1 to 10 parts per billion. So in this particular case, there's a bit of a derivation from the usual process of how GFoRSS would normally elaborate an MRL, in that the MRL would typically be tied to the health-based guidance value and exposure estimations. However, in this case, they decided to base it on the upper range of the analytical limits of quantification. So that's how they came to the 10 microgram per kilogram value versus the value of 590 microgram per kilogram, which would have normally been set based on the ADI that was established and the salmon consumption figures that they had available to them. So this is a bit of a detraction from the usual process, but I think which is becoming more prevalent in these codex meetings, they become more reliant on available monitoring data to show that a lower MRL essentially would have no impact on trade. So in this particular case, I think GFoRSS has demonstrated that, based on almost a decade of monitoring and a fairly large number of samples, that a MRL of 10 parts per billion can be readily met with the approved uses of diflubenzuron as something to control ectoparasites in farm salmon. So, and maybe if you want to, Samuel, you can describe some of the issues that might come up. But I mean, just to end, this is, I think you're going to see this more and more as the idea of considering other degradation products or potential metabolites of veterinary drugs and pesticides and food additives. And this is something that's typically done, but I think you're going to be seeing more and more of as you get more fulsome risk assessments that are being produced for various chemicals that are in food.
I don't know if you have any questions, any comments, any additional considerations that we need to take into account. Yes, Fadi, please.
I have a question, in fact, and comment for the salmon. Do there are studies for other type of fish? Is there an MRL for CPAS or for another type of fish? Or because the proposal for salmon only, so we can compare with the same food commodity, if there is MRL for the fish. And I also to confirm that the work with pesticide residues and veterinary drug has really been to be inconsistent. But I remember there is the same substances mentioned with 2 MRL, one in this Codex standard and one in this Codex standard. And in one time we faced the conflict. So we're trying to pose which one to follow. Just a comment. Thank you.
Yeah, thanks. I guess, Mark, if you want to take the first question, although, it's as you wish.
Yeah. No, it's a good point. There are a number of related vet drugs that already have approval for use in control of sea lice. And sea lice in particular tends to be an issue primarily for farm salmon. So as far as I'm aware, even though there are a a number of other MRLs for diflubenzuron and other food-producing animals. This is the first fish-specific MRL that has been requested, and that's primarily because there is already in certain nationalities ongoing use of diflubenzuron as a treatment for sea lice, but primarily in farm salmon.
So,
sure, but this does mean that we can apply this MRL in sea bass fish, for instance.
Well, that's a very good point, because I think this is part of the discussion that will be held at potentially either in past CCRVDF meetings or at this particular meeting in applicability of MRLs to other species.
Yeah.
So, to me, I would say that an MRL, especially based on the analytical method of detection, would be applicable to other fish species. But typically what GFoRSS then has to consider is residue data that might be available from practical use of that vet drug in that particular fish species. Unless a proponent can say that we consider, based on X example, say sea bass to be substantially equivalent to salmon, they could make that case. But typically GFoRSS would want to see some residue information from practical use.
Mm-hmm.
But certainly it's a good question in terms of, for one aquaculture fish, why isn't it applicable to other aquaculture fish? One of the main factors you'd have to consider is that the farming practice is near identical, because there's as with a lot of veterinary drugs that are used in aquaculture, there's a temperature consideration that has to be taken into consideration, so to speak, when you apply these drugs, because there's faster clearance with higher temperatures. So, but regarding your statement, I would say that there is a potential that you could be transferring, but a request would have to be made, at least for GFoRSS to consider that.
Yeah. So
because in this area we don't really farm salmon, we farm sea bass.
Sea bass, yeah.
Salmon, farm salmon always imported from outside.
Yeah.
I guess in support of that, there has been the use of diflubenzuron in sea bass, specifically for control of sea lice. So I think there's likely available information to do an extrapolation without a detailed assessment, but this would have to be something that either the CCRVDF or GFoRSS would then develop an opinion on.
Yeah.
For our area to collect this data, we need a recommendation, in fact, to test aquaculture products for pesticide. We have made this recommendation years ago, and that was on really some random testing happened for shrimps imported from Japan.
Yeah.
And we find some pesticide, but that was in a special request, not in a routinely basis. And the data is really very limited related to this. So if we can really recommend that, to have some surveillance for pesticide and fish, and to develop the method, because most of the organization in the area, they don't have the method to test pesticide in animal product or in animal tissue. More or less, they are focusing pesticide on vegetables and fruits or in agricultural products. But in aquaculture and hydro aquaculture study in farms and hydro, what they call the mix between agriculture and aquaculture, they are using pesticide in sea bass farms. So I think the area needs such surveillance, just at least if they are doing on every 3 years, one time at least.
Yeah. Thanks for that, Fadi. I'm going to go to Dr. Abdel Fattah and then to Dr. Suhair. So please.
Okay, thank you. Maybe I agree with Dr. Fadi that all the studies are done on salmon, and salmon is very, very expensive in our region. We don't consume salmon, but we have a popular fish like tilapia, sea bass, the catfish, and of course you know that even the different kind of fish behaves in a different manner to deposit chemicals in their bodies. And I hope we have other studies include other kind of fish with different behavior. Okay, and the other point I would like to focus on the European level, MRL level for diflubenzuron that decreased or reduced from 1,000 to 10 microgram per kilogram. This is very gap between 1,000 and 10, but this is not hazard. This is because the studies proved that diflubenzuron still have genotoxic effect, and this effect, even they reduce the MRL to 10 microgram per kilogram, still we have a risk of cancer, even at a rate of 1 person per million. Still we have a risk. So I hope this is to be considered. Okay, thank you.
Thank you, Dr. Abdel Fattah. Mark, do you want to address the genotoxicity issue, if you don't mind?
Okay, no, I think it's a good question. And I primarily, the direction that GFoRSS took was to challenge whether or not the 4-chloroaniline would be considered as a genotoxic carcinogen, or a carcinogen where there would be expected not to be a threshold. So in GFoRSS's current position, they've indicated that based on their most recent assessment, they would consider that there would be low doses of 4-chloroaniline which would not represent a carcinogenic risk. So there is always, we have provided in the document some different examples of other organizations that have looked at the assessment of 4-chloroaniline and their decisions regarding whether or not it might be considered a genotoxic or non-genotoxic carcinogen, i.e. a non-threshold or a threshold carcinogen. And I mean, essentially, they're coming out to the same conclusion. If you consider it to be a non-genotoxic or a threshold carcinogen, then you can define doses which would not be associated with a potential cancer risk. On the conversely, when you look at the decisions of the Committee for Medicinal Products for Veterinary Use, which advises the European Union and the US EPA, even if you do consider in a more conservative approach that the 4-chloroaniline might be a genotoxic or a non-threshold carcinogen, the cancer risk estimates that come out from this particular MRL are described as negligible. So essentially, this is a paradigm that's currently in risk assessment that by the time you get to cancer risk estimates of less than 1 in a million, the ability to actually accurately detect that cancer risk disappears. So it's simply, there could be questions about allowance of a genotoxic carcinogen in the food supply from a deliberately addition perspective, but
that's a
from
policy.
a toxicological perspective, I was actually a little surprised that they focus so much on the 4-chloroaniline, because in relatively high dose animal studies with the diflubenzuron, there wasn't any specific relevant information that suggests that either it was genotoxic or carcinogenic.
Yeah.
So we are really talking about trying to hypothetically extrapolate from very, very low doses of a metabolite or a degradation product.
I think actually we've done a good review of those two substances. I think for both cases there doesn't seem to be any concern in terms of moving forward with the adoption within the Codex process. There are some specific recommendations I think that we can highlight for the region. So on the basis of the comments that were made, we're going to be updating the draft report as it is. and we're going to make it available already on the website of the MENA Codex, so that it could be potentially used for the preparation of positions.